Hello, we are from Princess Julapon Science High School, Pathum Thani, Thailand. The title of our project is Study the Efficacy of Mya-TACF family extraction on inhibition of monoamine oxidase enzyme for development as a brain disease therapeutic drug.
Neurological diseases are more common in the elderly and tend to be higher. Common symptoms such as slow movement, trembling in a resting position, and tensing. So, we did a study and found that Parkinson's disease. There are factors caused by external and internal factors. It is internally involved in the release of the neurotransmitter dopamine. But in patients, dopamine is secreted too little. There is also an enzyme MAO which has two isoforms A and B. With breakdown neurotransmitter, there are current medications that focus on increasing the amount of dopamine and reduce the activity of MAO enzyme. But those still have side effects on users, such as dyskinesia and serotonin syndrome. So we want to find path in the treatment to reduce the side effects of current drugs. Studying the research found that alkaloid phenol and anthraquinone can also inhibit neurodegenerative enzymes. And in Mya-TACF that we are interested in, it consists of those substances and in large quantity of phenol. So we should to inhibit the enzyme that cause neurological disease. Want to study results of using Mya-TACF extraction in different pens and different concentrations to inhibit the activity of MAO enzyme, and 2. To study the efficiency of Mya-TACF extraction on inhibiting the activity of MAO enzyme compared to the standard solution.
For the overview of our methods, we started with extracting Mya-TACF family. Then we extracted enzymes from pig brain, and we measure the protein and activities of the enzyme. Then inhibit the enzyme by standard solution and by our extracts. And the last method is comparison and data analysis. For Mya-TACF family extraction, we use the 95% of ethanol and then we evaporate it. For extracting enzymes MAO from pig brain, we blend the pig brain and put it in a centrifuge. For measuring protein, we use Bradford test. And for measuring activities, we use fluorescence spectrophotometry. For inhibit enzyme by standard solution and extracts. For standard solution, we use cordyline or parthyline, and for extracts, we use our extracts. For the results, graph 1 shows the percent remaining activity of MAOA enzyme. The pink one is cordyline, and the lowest percent remaining activity of our extracts. You can see that the yellow one, rose apple at 100 microgram per milliliter, has the lowest percent remaining activities. And graph 2 for MAOB enzyme. The green one is parthyline. You can see that the yellow one at 100 microgram per milliliter, the rose apple, is also the lowest percent remaining activity extracts. For discussion and conclusion, comparison inhibiting MAO enzyme by Mya-TACF extracts with standard solutions. For the MAOA enzyme, cordyline at 0.08 micromolar concentration had the percent remaining activity at 2.19%. And the extract that makes the percent remaining activity of the MAOA enzyme had the lowest value is rose apple extract at 100 microgram per milliliter with percent remaining activity at 10.66%. For the MAOB enzyme, parthyline at 10 micromolar had the percent remaining activity at 77.33%, and the extract that can make the percent remaining activity of the enzyme the lowest is rose apple extract at 100 microgram per milliliter, which has the percent remaining activity of 11.17%. Therefore, the extract that can inhibit the enzyme MAOB and MAOA the most, considered at a 100 microgram per milliliter concentration, is rose apple extract. According to the research of Songkut Sarapusit et al. 2016, The plant with the lowest percent remaining activity had the highest inhibition ability. Thank you for listening.